Anyways, we're talking about H5N1. We're talking about a recent publication in Journal of Dairy Science. February issue 2026 Journal of Dairy Science. Journal of Dairy Science is open access, so any one person anywhere can log on and have access to your paper. The title of the paper is Exploring Influenza A Virus Receptor Distribution in the Lactating Mammary Gland of Domesticated Livestock and in Human Breast Tissue. And of course, I was interested, and then I got to the human breast tissue and I got really interested. Okay, now that's interesting. That's an interesting question. Fair enough? That's fair. That's interesting. Hello, and thanks for joining us for this episode of Dairy Digressions, the podcast from the American Dairy Science Association, the Journal of Dairy Science, and JDS Communications. I'm Matt Lucy, former editor-in-chief of the Journal of Dairy Science and JDS Communications, and a fellow of the American Dairy Science Association, and I will be your host. Dairy Digressions offers an insider's perspective on the science and the scientists shaping conversations both within and beyond the dairy sector. We highlight innovative research that advances understanding and fuels progress in the production and processing of milk for people around the world. We have two guests today for today's podcast on the H5N1 epidemic in dairy cattle. They are co-authors of a recent paper in the Journal of Dairy Science. Our first author is Dr. Rahul Nelli, research assistant professor at the Veterinary Diagnostic Laboratory in the Department of Veterinary Diagnostics and Production Medicine at Iowa State University. Dr. Nelli earned a BVSc and AH degree from the Agricultural University in India, his MVM from the University of Glasgow, and his PhD from the University of Nottingham in the UK. Dr. Nelli's research focuses on viral pathogenesis, host immune responses, and the development of innovative diagnostic tools to strengthen disease control. His work centers on influenza A viruses, coronaviruses, and animal populations, and other high-impact animal livestock pathogens, with particular attention to viral transmission at the animal-human interface and factors influencing host susceptibility and disease progression. Our second guest is Dr. Todd Bell, professor of veterinary pathology at Iowa State University, a diplomat of the American College of Veterinary Pathologists. Dr. Bell earned his DVM from Iowa State, completed a PhD in biosciences with a focus in virology at the George Mason University through the National Center for Biodefense and Infectious Diseases. Before his current academic position, Dr. Bell served in numerous leadership and scientific roles with the United States Army Medical Research Institute of Infectious Diseases. At Iowa State, Dr. Bell teaches systemic anatomic pathology while leading a research program focused on host-virus interactions at the cellular and whole organism levels. His work aims to better understand viral disease mechanisms and to advance the development of effective antiviral countermeasures. Welcome, Rahul and Todd, to the Dairy Digressions podcast. I got to ask you, Todd, okay, I'll just start here. You have to know my oldest brother is a retired Air Force colonel, okay? So how's it going from the Army to Iowa State? Tell me about that transition. We're going to digress for just a second.
Well, thank you for that question. And it's definitely an adjustment, and there's an adjustment period there. And I would say I think I'm kind of finally getting on the tail end of that adjustment period, but all in all, it's been fantastic. You know, there's pluses and minuses to the military and to the academic world, and being able to kind of merge those two worlds together, I think, and take the best of both of those and merge them together is a space that I'm kind of trying to navigate right now. But all in all, yeah, I think I'm finally getting to the point where I'm getting comfortable in this new setting and really, really enjoying it.
Are you two, how do you guys know each other? Do you, Rahul, I see you're a research assistant professor. How do you guys know each other? You just met each other at the water cooler, or how did that all happen?
Yeah, first of all, thank you for having us in your podcast. I met Dr. Bell at all virology symposium, which happens every 2 years in Iowa or in University of Iowa or Iowa State University. And at that first time I met him and I saw his presentation, I was impressed. And then that year, in 2 months, we had this outbreak of H5N1, and then we instantly connected together. And our research ideas aligned well, and his leadership is what I really admire. And then, of course, he's coming from all that military army background. So I see his leadership. So through his leadership, we navigated a much larger, complicated team, and we worked really well. And then we look forward to keep working like this forever. That's in short.
Yeah, I think it's interesting, this H5N1 thing. Maybe as virologists, you're not surprised at how tricky it is, but this is a tricky one. This is a tricky one when you have a pathogen like this that's mutating and traveling around populations. And, we'll get into it, but there's still, one of the biggest questions is how does it move around? Maybe it's not a question anymore. I don't know. But my limited knowledge comes from, we've done a few of these podcasts on H5N1, but I've also read about the same time your paper came out, there was a leading edge commentary article in Cell all about can H5N1 avian influenza in dairy cattle be contained in the United States. And so I kind of read that. They didn't have too many U.S. authors, a few U.S. authors, but not many. And I just thought it was interesting how much we don't know about this virus at this point, how much we don't understand. So I want to just back up and we're going to dig into your paper pretty hard, but I just want to ask you one quick question before we start digging in. And that is, any new updates, like really recent updates that you want to talk about in the situation in the United States? Okay, so let me give you my sort of broad look at this virus and kind of what's going on in the United States. First of all, we had as we all would agree, a very serious wave of this virus that went through the United States shortly after its discovery. Let's just say that. We've had a few little outbreaks here and there. I think recently we've got apparently a new, I don't know if it's a clade or what we would call it, this D1.1 that has showed up. And then we also have the situation in the Netherlands where apparently it showed up in a dairy in the Netherlands. I don't know how it got into the Netherlands, but is there anything, before we even start talking about your paper, is there anything sort of new on the veterinary front that you would like to share at this point that I sort of missed? Or what are you thinking right at this point?
Well, maybe I'll take a first crack at that and then turn it over to Rahul. But I think, to maybe quickly digress back into your last one of your last comments, which is this is a really complex problem with a lot of different angles and a lot is still unknown. And initially in the outbreak at the start of it in March of 24, I think back to also to where Rahul and I were introduced as the Associate Dean of Research here, contacted me in late March when we knew that there was a positive case here and the federal confirmation was being handled across town at NVSL, because our folks here at the diagnostic lab at Iowa State were to my knowledge, were the first ones to diagnose that. I think there were some other labs right on their heels to do that, but then the federal confirmation was at NVSL across town here, our partners. But as that whole process was going on, this 4 or 5 day process of that happening, Dr. Plummer kind of called me to his office and asked me specifically kind of in line with what you're talking about. This is a big problem. It spills over from wildlife potentially into livestock, could potentially go into humans. Who do we need to have in this large sphere to tackle this big and potentially impactful human health, public health, animal health problem? And so as a part of that, prior to this outbreak, had not done any flu virus research. In the Army, we did all kinds of biodefense stuff, but flu was generally the purview of civilian institutions. And so that's the first thing I let him know, that I'm not a flu expert. I am a virologist, but I'm not a flu expert. That being said, if you want to talk about the mindset that we would use in the military and how we would attack this as a team, I'm happy to discuss that. So that's kind of how I got involved. But probably one of the very first names that we brought up was Dr. Nelli, because of the 15 plus years of experience he has in dealing with flu and dealing with coronavirus and dealing with receptors. Because specifically, our question is, how in the world did this virus get into dairy cattle? And how did it get into dairy cattle? Flu virus is a respiratory virus. How did it get into the mammary gland? And where did it come from? So to your point, so many questions that require so many different types of expertise from different people. And it's just that team mindset that we needed right from the get-go to try to get ahead of this. And again, that's why we kind of brought Dr. Nelli in. To your second point, then I'm going to pause so Rahul can kind of fill in the gaps and kind of correct me where I miss things, is, what is new and novel? I think, again, going back to, we still have a lot to learn. What is the pathogenesis of this virus as it relates to other flu viruses or past bird flu viruses? And, how those two compare? Why is it getting into these different species? And one particular question that's really perplexing is, why is it so much more pathogenic in some species versus others? For example, cats get it, they're going to die. Cattle get it, they're going to get, the mortality rate is going to be really low. Some might get really sick. Birds get it. A lot of the birds are going to die. Some are not nearly as severely affected. So that piece of it is really kind of interesting as well. But I would say it seems as if, at least right now, that it's calming down a little bit on the dairy side of things. We haven't seen any cases, I think, since December 13th, maybe. If you look at the USDA APHIS website, again, that's kind of getting over my skis there, because that's definitely not our lane. We're the research lane. But that being said, again, I think that's a point that we keep coming back to, is the teamwork that is necessary to really understand this is phenomenal. And the final point I'll make before I kind of stop this rant is that it really was a team. Going back to the paper we're going to talk about, I believe there's 19 authors on that paper from Iowa State, from George Mason University, from the USDA, from multiple different departments here at Iowa State Vet, Microbe, Preventive Medicine, the D-Lab. I mean, none of this happens without all those people, and in particular leadership, allowing all of us to work together, because any one of those leaders could have said, nope, we need you to work on this thing over here. We don't want you working on this big project. But they were the opposite. They said, how can we get these people to work together across these departments and all these different areas? And they were really enabling it in order to get this stuff done. But Rahul, what did I miss there?
No, I think you addressed most of the things, but I just want to come back to the second question, right? What we are trying, what's new about this influenza? Yeah, this is, I believe all influenza experts would be, surprised to see that this virus is affecting mammary gland. So that was surprise for everybody. Nobody expected that. That's an unusual. And now we are looking into other organs, right? Whether uterus, rumen, urinary bladder, fetal, uterus, right? So this is, and as an influenza researcher, I always think like influenza always surprises us, and he has been doing it even before 1980 in the first Spanish flu. Before that also it might be doing, but we haven't had a recorded evidence. But now we are recording that. Now I see like it's happening. So why it's happening? And then as you said, how in the hell they got into Netherlands? Because this is all about the birds, because influenza is always inside birds. Birds keep on carrying this virus, and then occasionally they spill into other animal species. In this case, it's mammalian species that includes even marine mammal. And if you look at the current, stats and numbers, there's so many sea lions and many other marine mammalian species got also infected. So we are talking about more than 200 mammals got affected just because of this new strain. So hence, this is surprising for everybody, and influenza always does this. It's not that's not what I'm surprised, but why it got into this first place is what we're asking the question right now.
So, Rahul, I know you worked on SARS-CoV-2 and white-tailed deer. Is this more or less confusing than SARS-CoV-2? Is it more or less confusing than that virus?
I don't want to talk. That's a tricky question. Is it corona or influenza? People in fact, all influenza experts worked on corona at that time during COVID, right?
Okay.
I feel like influenza is, if I have to prefer, I prefer influenza. That has the surprise element all the time.
Okay.
So corona probably it'll be again, these are happening in the birds, in wild animals all the time. It's only when it affects humans, that's where we actually get, anxious or maybe start panicking about those things. But these events are happening, but we're not doing enough surveillance.
Yeah. So like SARS-CoV-2, it was pretty much, it was a respiratory thing, I think. I'm not I'm showing my ignorance here, but it's not like it, like this one kills cats, different from a cow. It doesn't kill cows, kills cats, kills birds, but the bird killing is different from the cats, from the cows. That's totally messed up, isn't it? Or is that normal for these viruses? They just do whatever. They kill whatever way they can.
One common finding, at least what I see in all these incidents is this pro-inflammatory response. When animals overreact to these pathogens, that's in most of these scenarios where you're seeing death is associated with active cytokine storm, we call it. Whether it's SARS-CoV-2, that's what we are all got. To begin with, most of us got infected because of cytokine storm. That's the causing the disease. And so is influenza. And I classify H5N1 at least, right, in my view, post-COVID and pre-COVID. The pre-COVID numbers of H5N1 in humans is 50% mortality, right? Post-COVID, in this case, we have 71, and the mortality is less than 1%. So because the humans who got affected, there's only 2 deaths out of 71 cases. So if you do the number, because when I'm doing my PhD, there was almost 50%, and that's what the panic is all about. Okay, this is a nasty flu. If it comes, we're gonna be prepared. So in those sense, so I feel it's not necessarily the virus. The consequences is most about our immune system overreacting. Why is it reacting like that? Is what I wanted to understand. That's my whole research theme.
So a couple of things in those comments you guys made, I just want to touch upon. first of all, Todd, I love the concept of bringing that military experience to a question, because the military is, known for its discipline and sort of getting things done. And I think in focus, you have an objective. Let's do this. I'm not a military person, but, my brother is a colonel, retired. So I could talk about military. Oh, I took a lot of orders growing up. But anyways,
and an older brother, no less.
Yeah, no older brother. I actually have a niece who's a flight surgeon, and she's in Ohio. And then I got a nephew who's also, he's a military guy. So, but it didn't happen for me. But I like that comment you made. I thought it was good. I thought that was really it. I thought that was just super interesting. And you both brought up the importance of building a team. And I noticed that when I looked at your author list here, is a big team, and it also goes across organizations. You've got USDA people here. You've got Virginia Espina from George Mason, excuse me. That USDA Animal Disease Center, that's pretty nice to have down the road, isn't it? To have the magnitude of that. Is it tradition? I think it's always been the tradition for Iowa State Veterinary School to have some relationship with that group. Is that true, or must always have some relationship with them?
To my knowledge, yeah, it always has. And a lot of folks are trained here and work there, or maybe do some postdoctoral work there and eventually come back here, or there's some sort of a collaboration. And so the geography naturally kind of brings them together. In addition to that, folks have just, known each other for so long. Institutions that, so generally they've worked together for.
It looked like I was
quite a while.
Yeah, I was looking at some of the people, and I noticed that I think there was a postdoc that was there, but is now with Iowa State, I think. And then some of these co-authors, like that Bailey Arruda and Amy Baker, they're pretty heavy hitters in this world, I think. Is that right or wrong?
No, it is. And kind of funny, so Amy Baker and I were in the same vet school class of 2002.
Oh, fantastic.
Yeah. And so, and we were, I would say we were friends in veterinary school. And, at least I know I had respect and admiration for her and have kind of at a distance followed her career over time, but we hadn't really spoken, face to face in 20 years. And then this happened. But, we had such a fun vet school class and we're so connected, we could pretty quickly pick up that phone back up and start talking to one another, working together. So to your point, she's both a heavy hitter, but it's really nice to have that personal relationship ahead of time in order to bring that team together. And Dr. Arruda as well is a really, really sharp individual and is a heavy hitter as well.
So I think, for you listeners who are wondering, what are we talking about? Well, now I'll tell you. Anyways, we're talking about H5N1. We're talking about a recent publication in Journal of Dairy Science. February issue 2026 Journal of Dairy Science. Journal of Dairy Science is open access, so any one person anywhere can log on and have access to your paper. The title of the paper, Exploring Influenza A Virus Receptor Distribution in the Lactating Mammary Gland of Domesticated Livestock and in Human Breast Tissue. And of course, I was interested, and then I got to the human breast tissue, and I got really interested. Okay, now that's interesting. That's an interesting question. Fair enough. That's fair. That's interesting. So I had a couple of questions I just want to ask you. So basically, we're looking at a paper. You're looking at receptor distributions. And just for the audience, one way to understand where maybe I'm wrong, but I'm just going to summarize for the audience. One way to understand where a virus might infect or a location is to sort of look for where that viral receptor is, the tissue that expresses the virus. Is that correct? Is that one way you guys do that sort of work?
Yeah, no, and I think we generally try to use a simple analogy to explain the concept that you're referring to, and specifically that the virus and the receptor it needs to bind to is kind of like a lock and key mechanism, where if specifically that virus, which is the key, and the receptor is the lock, fit together, then that virus can attach and then get inside the cell. And the viruses, by their nature, are parasites. They have to get inside of a cell in order to reproduce and continue their life cycle. So that virus with lock and key mechanism with the receptor has to happen. And so specifically, our really interest was, one, there's a mammary tissue of cows. Is that how the virus maybe got in? Are there the virus, which is the H5N1 in this case, are the right receptors, right? Or the right locks on those cells for that virus key to fit? And we looked at the mammary glands of those animals and found out that was the case. And so then that got to thinking that was the first paper we published. And the second one we published, which was in the Journal of Dairy Science, was, well, if cattle can get affected, what about all these other species? Do we need to be looking in milk of these other species to make sure that milk from these animals is not showing up somewhere where it might be infected as well? And so specifically early in the outbreak, it had been reported that alpacas have been infected. It had been reported that goats had been infected in Minnesota. And then there are certain reports out of England of sheep being infected. And then, as Dr. Nelli mentioned, 71 human cases. So we thought, all right, well, let's look at all these different species that we know can be infected, and then potentially are the receptors for that virus in those areas. And so could it, potentially show up there as well? That was kind of the thought process behind that.
Yeah, so that was, you guys looked at pigs, sheep, goats, camels, and humans as well, which was interesting. And I have a sort of a little bit of a digression here. And as I read your paper and you guys went through all the different receptor types and stuff like that, and we're talking about influenza A virus. Everybody talks about AI and the ability of AI. This is not artificial insemination. This is like artificial intelligence to predict stuff. Like, if you just fed all this information into some supercomputer, would it have predicted this outbreak? Would it have said, well, you dummy humans, we got these viruses and they do this, and these cattle do that, and they have the receptors, so it's just a matter of time before this is going to happen? Would a supercomputer ever predict what we're dealing with right now?
Well, I think part of what our initial research was to find out there had been nothing published on there being receptors in mammary glands of cattle. So I think oftentimes those AI systems, if I understand them correctly, which is, I have a very rudimentary understanding of that, but basically it's scanning the information that's out there that is available, and then it collates all that information, but that information would not have been found there. Now, that being said, there's some methods where I can kind of learn and do some of those things, but so to that question, I don't know. That's a good question. Rahul, what do you think?
No, I would definitely say no at this point, because that information was never available until now, right? So the AI models, we need to teach them what to look for. That's because they're not smart. They're actually gathering all that information, digesting for us, and then giving us that simple one-liner statements. So I think we did this now. Now the AI models can predict based on it. But indeed, we are actually working with our AI experts here in Iowa State too. So how can we do these kind of predictions? So for doing those kind of predictions, you need to feed in a lot of data, and a lot of data we're talking. And that's what we as Iowa State researchers, many of us are working in those lines to give that data to those AI scientists such that it can be predicted. I don't know.
Yeah, that's good. So that gives me hope. I mean, we actually still have to do research. Wonderful. I mean, we actually have to do something in our laboratories. A message to you young people, there is work to be done, right? So when you make a new discovery, and that's awesome. I'm glad. Yeah. I'm glad. Rahul, the one thing that I saw on this paper as I was looking at it, you didn't do white-tailed deer. I thought you were a white-tailed deer guy. No, none of this in white-tailed deer, or what?
I don't want to open another Pandora's box. We already enough of that, right? I don't want the hunters now to start coming to me and say, hey, now what next, right? So no, jokes apart, right now I think at one point we were thinking at it, but at this point we don't have that mammary tissue of them, but we do have those tissues. Probably we might look into that, or maybe it's the next paper which is coming out. So just
hold tight.
Yeah, hold tight. Yes.
One of the things that, again, for our audience, these two investigators, the way they approached this problem is took these mammary tissue from these different species and looked for specific receptors. And the couple things I want to talk to you about. So just as an aside, in reproductive biology, if we wanted to look for a specific cell surface molecule, we'd raise an antibody and we would do something called immunohistochemistry. Okay. And you guys are doing something called lectin histochemistry. So realizing that a lot of the repro people never heard of this term lectin histochemistry. We thought it was a typo. I'm surprised the reviewers didn't ask you about it. But can you just tell us a little bit about these receptors you're looking at? Okay, I noticed there's this alpha 2,6 and alpha 2,3 or whatever. And also explain the approach of lectin histochemistry, because that's not an antibody, that's a lectin. And so I've read, so I understand it now, but tell us a little bit about that process you use to identify these things?
So, lectin histochemistry is not new. It's been there for quite a while, but basically we are in, as your Repro, what you're saying about IHC, there you're looking at an antibody. Here, a protein molecule. Here we're looking at a sugar molecule. So do you have that sugar molecule or not? So the sialic acids are nothing but sugar molecule. These are monosaccharides, right? So we are identifying if those are present or not. And in fact, they'll be also present, plenty of them in the uterus, if you're thinking about Repro. But what we are doing is we actually, in order to identify those sugar molecules, we need to have equivalent, binding molecules. And those molecules are these plant lectins. So Sambucus nigra and Macchiambrensis lectin, these are isolated from certain plants. And then some of these lectins can also be isolated from snails, from oysters, there's different. So they interact with these sugar molecules and then say, hey, and then they attach. And the moment they attach, we now tag them to a fluorophore or hydrogen peroxide, such that we can actually look at a color. So those fluorophores, in our paper, we looked at green channel, red channels, blue channel. So basically the red meaning, with the red fluorophore tag to it, same as green is FITC. So in that sense, right, influenza has, predominantly uses these sialic acids called sialic acid alpha-2,3 and alpha-2,6. Alpha-2,6 is traditionally known that mammalian, meaning the influenza viruses which originated from a mammal, typically bind to those receptors. And if it's avian, they bind to 2,3 receptors. If the virus originated from avian, that means chicken and duck. And in more detail, they actually have 2,3 as subclassified from duck origin and chicken origin. So if it's a chicken origin, we use a lectin called MAL1. And if it's a duck origin, we use a MAL2 to see if those receptors are present. And MAL1 goes and bind to sialic acid 2,3 receptors, which has N glycans, and MAL2 go with the O glycans. So hence there is a differences in these sugar molecule structure. So that's what we are looking for. And if both are present, then there's a concept of called mixing vessel. If on the same cell, if you have both 2,6 and 2,3 receptors, then there's a chance of avian and mammalian viruses infecting the same cell and creating a mutant virus.
So that's what we don't want. We don't want that, right?
We don't want it.
That's correct. I can't remember a little naughty secret. I'm colorblind. Okay. So you're killing me with this paper. All right. You're killing me. All right. Killing. There's all this beautiful fluorescence, and you've also got some different chromophores, but you got a lot of beautiful fluorescence. You're killing me. Okay. But can you just tell me, did you see evidence that they were present on these two receptor forms were present on the same cell types, or was it always one way or the other?
We did find them in some, not always in the same cell, right? But there's a lot of variation, right? So in some areas we saw both are present, co-expressed. In some areas there's only one receptor expressed, right? So, but the point is how likely the virus is going to bind to that tiny area where they have these receptors. So we don't know yet. And that's the part we want to do. And is there a relevance like the abundance of these co-receptors? So is 2,6 more abundant or 2,3 more abundant? And do they have any affinity? If there's more abundant 2,3, the virus is going to go and bind, so those kind of studies we haven't performed yet. So that's in the pipeline. But based on the literature, what we know is there is a high chance if both receptors are present, there's a high chance there's a possibility for both avian and mammalian viruses infecting the same cell.
Okay, so I keep bringing up the cats, and I apologize for that, but they seem to be like the canary in the coal mine on a lot of this stuff. But do you think if you look for cats, is it that the cats don't have these receptors in the mammary gland, or is it that they have these receptors in the central nervous system where other species do not, or is it some other anatomical or biological situation in a feline that really differs here? And I guess just to totally digress, our wildcats, not domesticus, but our pumas and mountain lions and those species in North America, are they affected similarly, just out of curiosity? So there was a couple of questions there. I apologize, but
no, my guess is yes. Most likely they might have these receptors, but we haven't looked into those pumas, mountain lion. But in domestic cats, yes, there is evidence that these receptors are present, and more so in the brain. Where there is more of this infection, the brain is abundant with these 2-3 receptors.
Mm-hmm.
So most likely, that could be linked to, hence the virus is, replicating efficiently in those tissues in domestic cats. But to even do any such studies in wild cats or collecting samples, there's a lot of paperwork and regulatory mechanism to even collect tissues from those wild animals. It's hard. Yeah, as and when, if we have any of such those, yes, we are happy to look into those and then see what's going on. Yeah.
And to that end, there has been some reports in the literature of some of these cats. I think I'm struggling here. I just read a paper this morning or yesterday about some fatal, highly pathogenic avian influenza in leopard cats in South Korea. So these animals that predate on these birds that die from high path. So it's not only domestic cats, it's some of these other cats that are looking for food and they eat these birds that are dead from it and they get it as well. And then reading some of these papers, it looks like the pathology is fairly similar. So cats in general are very susceptible. In prior outbreaks in Southeast Asia, sometimes there have been outbreaks where they fed some of these birds that died to even wild animals in the zoo, and there's been kind of similar issues.
As I talked about these cats, and I understand, wild cats, I was reading Science this morning, and they said, and they were talking about this issue of getting samples from wild species, and it's going to sound a little odd, but it was Science magazine. They said, there was an article in there that, certain investigators, some of these species are so rare that if you happen to hit it with your car by accident, that death can actually mean something scientifically. So I'll just put it out there. If you guys hit a mountain lion, you send it into Iowa State, and I'm sure they'll take it. Just keep it cold, right, Laurel? Keep it cold. Okay, keep that cold. Send it in. Look for his address. It's in the Journal of Dairy Science, I'm sure, But it was really interesting. I never really thought about that. But some of these species, you just don't get samples, And so they were bringing that up, and they were going through all anyway. So I was interested in your work with human mammary gland, and I understand that you mentioned in your paper those were a little bit different kind of sample. I don't think it was fresh tissue. For example, you got it from a human mammary repository type thing, a little bit different, but you did see evidence for these receptors in human mammary tissue. Am I getting it correct when I say that?
Correct. Yeah, and that was our collaborator from George Mason that they we collaborated with them, and we went through all the proper paperwork, which you might imagine there's quite a bit of paperwork there associated with those samples, but they were very old bioarchive samples that had kind of been put to the side for some time, but we were able to take some of those specimens that were de-identified and just look in general at some of that tissue that was from, I believe it was probably a kind of a cancer trial, and look at those tissues to see if both the 2-3, which is again the avian, and the 2-6, which is the mammalian, blue receptors were there, and we found both present in those tissues, even though the tissue had been archived for quite a while.
So from a One Health perspective, okay, and so I know from One Health perspective, okay, if a mother was infected with this virus, for example, unknowingly, and she had it, or maybe she didn't know it, is it possible that she could, if she was nursing, that virus could then be transferred to her infant? Is that in the realm of possibility here?
Well, and again, I'll let Rahul comment on this after I'm done. But I think so. If we look at a comparison, do some comparative pathology here. I mean, look at cattle, and you look at the viral loads present in cattle, and the overall viral loads in the mammary glands of those animals is astronomical. And how the virus got in there in the first place is still unclear, but it's very clear that it can replicate to very, very high levels in the mammary gland, and that it comes out in those very high levels in the milk. And so because we don't know that about cattle, we've studied even less this problem in people. How would it get in there if there's a very contaminated environment, like in a milking parlor, if there's a whole bunch of infected milk that got splashed on clothing somehow? I don't know. How that would potentially get there, or if they got it via respiratory route and they could travel to the mammary gland. Again, I think that's an open-ended question whether or not that could happen. But that's a great question. But again, just even in general, with it showing up in dairy cattle, it's like, well, let's look at humans then and see if because we don't know how it got there in the first place. Let's see if the receptors are there. But Rahul, what do you have to add to that?
No, but that's exactly what I would have started, but I would add more actually. Yeah, we don't know, and we don't want to get into that debate about One Health. It's a very controversial, because there's no evidence as such of today that it can happen. But if it happens in dairy cattle, I would be open-minded to think it can happen even to infants.
Do any of the...
There's a chance because these receptors are present right from a very young age, So there's a chance of these sugars, sialic acids, present in humans, and the chance, the virus can bind, but have we done any of those studies yet? No, we haven't. So hence we can't directly link it. But at the same time, we should have an open mind and start looking into those directions and start researching in those areas and see if we are prepared for any such scenarios.
So I tell you, you talk about One Health. I'm a big fan of the TWIV podcast, which is This Week in Virology, and that is human virology. And I tell you, they talk a lot about H5N1 in dairy, and you'd be amazed how those guys talk about H5N1 in dairy and poultry and implications for human health and stuff like that. And so you just get the sense that this is on everybody's radar. This thing, this H5N1. Yeah, I don't think we ever have any documented cases of someone drinking a glass of milk and getting H5N1. I don't think those really exist. I don't think those exist. There certainly has been some, I guess it's human to animal transmission. I guess there's been some of that. You mentioned 70 cases. Is that this year, Rahul?
Yeah, since we had it from 2024.
70 cases.
71 precise, yeah.
And I didn't quite understand when we started talking, you said that this virus used to have a 50% lethality rate in humans, but now it's far lower than that. And I didn't quite understand why that was. You mentioned SARS-CoV-2, and I wasn't really sure.
Yeah, no, I think that's because the current circulating H5N1 is from a different clade than which is the original H5N1. So they are different. Even our PCR testing wouldn't recognize the current one if we use the old PCR.
Yeah. Okay.
So things have updated. The virus have evolved. The virus has changed. So that's probably why. And there are not that many mammals got infected earlier.
Mm-hmm.
It's the first time we are seeing these many mammals, like a quite range. We're talking a wide variety of mammals getting infected.
Mm-hmm.
So hence, I believe that could be associated with virus evolving and mutating, and then targeting new tissues or new regions of new animal species, new tissues in those animal species. So yeah, that's influenza. It does that.
So is this is influenza A, and that, I'm not a virologist. This is a little bit like human flu virus type stuff. Is it the same thing?
Yep.
So,
but bovine has influenza B and C and D.
Okay.
So that's different from influenza A.
2 questions. Does my annual flu shot as a human provide me any protection to the bovine virus?
Sorry, you mean the current circulating influenza A?
Well, if I got a flu shot from my grocery store this fall, or last fall, is there any efficacy against the bovine H5N1?
Okay, so as per CDC, they mentioned that there's some level of cross protection from the flu shot we get, but how much and how much dose you are getting, or it depends, right? If you have more dose, that again, nobody has done those kind of studies yet, or maybe people are working on it as we speak.
Do you foresee a cattle vaccine? Is that a solution to this problem, or is that needed? Or is that we can manage this thing in other ways? Do you see a cattle vaccine, number one? And number two, if this thing is constantly mutating, are we going to have to change that cattle vaccine every year the way we change the flu vaccine every year?
That's a great question, but I would be very diplomatic in my answer. I don't want to, be one way or the other, but we need to think about the cost dynamics as well, right? So how much is it for the farmer to have this flu shot every year? Is it going to add additional costs? And all those factors you need to build in. Then you talk about a vaccination strategy. So is that going to eliminate? Is it causing a disease, what do you want to avoid? Is it the disease you want to stop, or is it spreading from into humans is what you want to stop? So those are, if you build in all those components, then you should have a clear answer. Why do you need vaccine? But if you just go by, okay, because we are having flu shot, cattle should also have a flu shot, that doesn't make sense to me.
Okay.
Because we live 70 years. I don't think most farms, the cattle doesn't stay for 70 years. I'm just saying world average of age expectancy.
So it's interesting. So, and to some extent, it's just so early in our understanding of this thing that we can't even make logical predictions. So I have a question, and I want to just touch upon what a dairy farmer might be able to do. And I know you guys are basic scientists, but what is the current sort of Iowa State recommendation for dairy farmers? I mean, do you guys have a list of things you say to them that you need to be thinking about or doing to prevent your herd from being infected? I mean, obviously, we'll take off the table, don't buy cows from the state that's got a raging thing. We'll take that off the table, because that's obviously, I think when we think about how this thing moved around, the first and foremost, it moved around from people moving cattle around, which they didn't do that on purpose. They weren't trying to do anything wrong. It just was the way it was. But what about if you take that off the table? Is there anything you can do to stop this thing from moving around? I heard anecdotally from California, it was like, well, if your neighbor gets it, you're going to get it. Okay, so just hang tight. But is there anything that you see, from your expert perspective that keeps this thing from moving from into your herd or nearby?
I mean, if you ask me my own personal view, nothing associated with Iowa State, right? So the way biosecurity measures would be key and critical. And in Iowa, we are on that fly zone, that wild bird fly zone.
Yeah.
Flying from north to south, south to north, right? Hence, we are seeing, even in January, we saw a lot of dead birds. The Canadian goose dropping dead from sky, in the lakes around it. So those are the things you need to watch out on your farm and see if those dead birds, take it for testing and make sure that it's nothing to do with influenza, right? And don't allow your cats and dogs, to either play with those dead birds or maybe try to eat them. So those are some things which we can all do collectively to contain it using those kind of biosecurity measures. And then as we are doing this, we are actually looking at how most researchers are looking into this. How did it get in the first place into the mammary gland? So by experimental study shows that it's only through mammary gland you are having those high virus productive virus. Productive virus means it goes and infects another cell. Just because you have RNA doesn't mean anything to me. You need to have a productive virus to go and infect. And that is happening mostly in the mammary gland. But even in the experimental animals, when they give through respiratory tract, you're not finding the same levels as you're giving through intramammary. So there's something in that mammary gland. I'm super, super excited to work in that and then understand what's going on there and what's not going on in the respiratory tract. What's the difference? That's what I want to understand. So as we are doing that, if the farmers and all our stakeholders can do those biosecurity control measures and try to contain that for the time being, probably we can come up with mitigation strategies, maybe a drug, maybe a vaccine, which you don't have to do it every annual year. I don't know, because there's so much research going on right now.
Yeah.
Well, and maybe if I could add that a little bit, I think to Rahul's point is biosecurity is a key. And then two, I think you mentioned it as well, we're the basic science and we try to help with the research side, but we would point folks to the USDA and APHIS websites because they have some great information on biosecurity, some great people you can contact through there. We would have folks contact our extension veterinarians here to help with that piece of the puzzle as well.
Mm-hmm.
They do have some great biosecurity information there. And then again, that's really what our goal is to get the questions from the regulators, the questions from the farmers, and what are they seeing, and put it all together and then formulate that to find our next research question and then try to smartly design our experiment so whatever scientific output we get can help those regulators or help those farmers make biosecurity decisions to protect their farm. And so that's really kind of what we hope to add is that piece of it.
It's just such a mystery. Is it the wind? Is it the birds? Is it the fomites? Is it how does this thing move? And then I hadn't thought about till Rahul said, but how does it actually get into the mammary gland? Is it coming up through the teat canal? Is it some other route? And I'm not sure I can get this out correctly, but no one wanted this to happen. But as scientists, it's fun to work on an exciting problem and a challenge, right? But I'm not saying that we run around hoping something bad's going to happen, but it's exciting to work on a challenge and to contribute to solving a problem as a scientist, and so this is a very exciting problem. It's an exciting area of research. I have this quote, and I got this from TWIV, and I got to read it. It's from Louis Pasteur, and it says, the role of the infinitely small in nature is infinitely great. The role of the infinitely small in nature is infinitely great. I'm not a virus guy, but I started doing a little bit of microbiome work on the uterus, and you gain tremendous respect for the infinitely small when you start working with viruses. Fair enough?
Yeah. Well, and we appreciate you, kind of fleshing that out, because we feel the same way. And then one point that I would like to maybe highlight regarding what you just said is the entry, right? How's it getting into the mammary gland? And then how we designed our experiments was to look from the distal aspect of the T canal all the way up into the mammary gland, so we could hopefully kind of start to answer that question. Did it come from down, from top and go down in, or did it come from the bottom and go up? And specifically looking at the receptors in all those different areas, what is possible then? If it was in the environment and these animals are laying down where these geese have just, deposited a lot of their materials, and the teat is right in that spot, is that a possibility? Or did they breathe it in and then it got in systemically? So again, that's kind of thinking about what they're seeing on the farm really helps us design our experiments, and then helps us look at the right things. And so again, just that whole One Health, Team Health, everybody trying to get in the same room and think about this together. I mean, again, I think you're obviously a scientist, just the way you're processing this information and asking those questions, because that's what we really try to do as a group, is to ask those questions, and then in the hopes that we can design our experiments appropriately to answer those questions.
Yeah, that is, we're going to have to wrap up because we've taken about an hour of your time, but I wanted to ask you, was there anything else about this paper that, you would like to highlight or like to mention at this point before we wrap up? I mean, is there some aspect that maybe we could have discussed a little better or talked about a little more? My summary of the paper, my understanding is you look across a variety of species and you use these technologies to determine whether receptors were present, and you found that in the mammary gland across these species, you're seeing the receptor. Now, we don't know, I think it's fair to say, we don't really know if this certain species is infected, whether or not they will shed the same viral titers that a bovine will. Had a few podcasts. The viral titers are astronomically high, I believe. Like, whoa, scary high. And so anything else you'd like to mention about this paper or thoughts about the paper?
Now, one thing I would like to add is this, we mentioned about your speaking to so many virologists or One Health experts. So considering these kind of studies, those are expensive to work with. And we know the microphysiology or physiology as a veterinarians, as researchers in the veterinary field. So I would encourage the human virologists, right, to work closely with us and help us support that funding, which has to go through this kind of work. It's not as easy as to work with mice, in fact, I'm not saying that's easy, but I'm just saying we have a much challenging work here to do this kind of work. So hence, working collaboratively with veterinarians and vet schools and funding those to do this kind of research and would help address the real world problems. That's what I want to say.
Well,
fantastic.
Yeah, and I would add just one point. It's just that the need to be vigilant with respect to looking at this human-animal interface. And because it looks as if all these different species could potentially support infection. We didn't look at replication. We just looked to see if the virus could get in. But then the next step would be it can replicate in those tissues, right? So the sheep, goats, alpacas, pigs, humans. So that's a lot of different species where this could potentially show up in mammary glands or in milk. So being vigilant with respect to looking at and surveying this human-animal interface to make sure it's not showing up either in that spot specifically or in other spots in general.
I want to ask you a few kind of just departing final questions. And Todd, I'm going to ask you first. Your passion for science is just crazy, both your passions. But if you weren't a scientist, what would you be doing? What would you be doing if you were not a scientist?
Yeah, no, it's funny you should ask this. And then I guess I'll kind of let you in on the TWIV reference you used earlier. Actually, I was on TWIV when I was in the Army, and they asked the same question. And so my answer then, which would still be the same now, is I had aspirations of playing shortstop for the Chicago Cubs until I was in my 40s. And then doing something like this, my lack of athletic ability and size and speed in general prevented that. So I just went straight this route. But, if I had the athletic ability, I would have played shortstop for the Cubs.
Fantastic. So, Rahul, do you ever think about doing anything else but what you're doing right now? I mean, you wanted to play cricket, right?
Yeah, I did it already for my university back home. But if I have to, I would either choose, go back to our shrimp farming. We have aquaculture and do some shrimp farming or be a politician. Those are the two contrasting,
I tell you guys have been great. And I wanted to thank you so much for this engaging and insightful discussion. It's been excellent. We've done a couple of these sessions on H5N1, and this was entirely different, which really makes me happy. So it's not going to compete with nothing you said competes with what we've already talked about on this one. So that's just makes it really special. We truly appreciate your sharing your expertise. And I also appreciate your time, and we appreciate your submission to the Journal of Dairy Science. I always get really excited when I see a great research paper. So thankful you didn't send it to the Journal of Virology. If there is such a journal, I don't know. But anyways, probably. But we do appreciate that. So, and thanks to all of you for joining, listeners, for joining us for this episode of Dairy Digressions. If you enjoyed the conversation, please like, subscribe, and rate the podcast, and consider sharing it with a colleague or friend. We always welcome your feedback. Feel free to reach out to us at adsa@adsa.org. For show notes, past episodes, and updates to everything happening at ADSA, visit adsa.org. And until next time, thanks for listening to Dairy Digressions. Thank you, too. Thank you very much.
Yep. Thank you.
At Dairy Digressions, we talk to the people behind the latest discoveries in the dairy industry. If you would like to connect with those people, then the American Dairy Science Association is for you. ADSA is where researchers, educators, producers, and leaders in the dairy industry come together. Join us and become a member today at adsa.org.